Okay, it is 10 o'clock on my watch here. It is my great pleasure to welcome Professor Laura Reed today with us. Professor Reed is Associate Professor at the University of Alabama, Department of Biological Sciences. She did her PhD in Ecology and Evolutionary Biology at the University of Arizona in 2006. And did a postdoctoral research at the North Carolina State University. Professor Reed is heavily interested in using animal models to study diseases. And well, many people agree that animal models are helpful. Most people think about, rhesus macaques or mice. And Professor Reed says, well, you can learn a lot from Drosophila because it's cheaper and it's smaller and it's faster, I guess. And maybe there are more advantages that Professor Reed will tell us. So she is using a Drosophila model for metabolic syndrome. And her primary research uses systems biology to correlate empirical data from across physiological levels, such as RNA metabolites. But she's also really interested in evolutionary genetics of speciation. Because she thinks, and I heavily agree, that ecological context is essential to understanding the evolution of genetic architecture, and I might want to add metabolism. Metabolism and specific metabolites are often specific for certain species. And in my past, I was a plant scientist, and that's definitely true for plants. It is less appreciated for other organisms, but I'm pretty sure it's certainly true for other organisms as well, including bacteria, of course. So with that, I look at the number of participants. We have still some people coming in. Again, welcome to the UC Davis West Coast Metabolomics Center monthly seminar series. My name is Oliver Fien. I am a professor here, director of the center, and I welcome everyone to join us to this online seminar. We see that we have guests from across the United States. Steve Barnes is a frequent guest of ours, also from Alabama. I see his face here because he turned his camera on. Other people have welcomed us in the chat room. So yes, please take the chat room. You can certainly interact during the seminar. We will collect also questions during the seminar that we then read to Laura Reed after it. Arpana Vania, our outreach coordinator, is waving at you, waving back. We'll collect these questions and then sort them into topics, which is clearly a benefit of having online seminars. And I'm sure that when we are all vaccinated next year, we are all looking forward to that, that we will keep online seminars because it's a great way to reach our large audiences. And actually, even discussions are a little easier sometimes during these online meetings. While certain things like the dinner and the show and tell and the interactions with postdocs are missing, some aspects are better. All right, so I think without further ado, I can switch off myself and welcome Professor Reed to give her lecture.
Okay, so unless I hear otherwise, I'm going to assume you can see the slides. Let me know if you can't. So as Dr. Fien suggested, I'm an extreme enthusiast of working with model organisms, especially small invertebrates, as a way to model disease in, for human disease as well as other basic evolutionary questions. So I'm going to start out with the context of, which is well recognized among this audience, of the relationship among the genome to the phenotype, that there are many sort of intermediate or endophenotypes that connect the genome to the phenome. So there's the epigenetic, transcriptomic, proteomic, and then of course for this audience, the metabolomic realm between these levels. And then all of these have regulatory feedback among the levels that ultimately produce a phenotype. So in most research studies, we tend to sort of just take this as the basis or starting point for our analyses. The issue is that we're often neglecting the variation in phenotype that occurs naturally in a population, and how that variation can be induced by environmental factors. So what I personally am truly interested in is how does natural genetic variation and environmental variation ultimately produce phenotypic variation as mediated by these endophenotypes. So there are many different potential ways to approach this question, and I'll show you a couple of the ways that I'm thinking about this in my work. So my work is primarily focused on metabolic syndrome, so obesity and its comorbidities, because they have both genetic and environmental influences, and then are obviously also of major public health interest. We've seen a dramatic increase in the rates of obesity in the US just in the last 10, 20 years. Where I'm here down in the Southeast, we're not doing so well in terms of our rates of obesity. So it's definitely a serious public health concern in this area. But so there's been a transition from low levels of obesity, relatively low levels of obesity, to much higher levels of obesity in a very short period of time. So this is indicative of an environmental transition. There have not been dramatic genetic changes over that time period in the US that would explain this pattern. So this is an environmentally driven change. But despite that, we also know that there are thousands of genetic loci that contribute to risk for obesity in humans. So at the point that I pulled this number, there is nearly 3,000 different known associations in the human genome with obesity traits. So what this means is that there's both genetic factors and environmental factors, and each gene only contributes a tiny proportion of the total genetic variation in the trait. So we can't necessarily expect to solve all our problems by finding the genes. So I'm going to talk a little bit about why I work with flies, and then transition into looking at some of the work that I do with those endophenotypes leading from genotype to phenotype, and then some of the genetic mapping we've done to try to identify some of these genes that might be mediating some of these relationships. And finally, I'm going to finish up with talking about some of the other environmental factors that my lab is working on. So initially, I'm going to focus primarily on diet, but we're also thinking at other levels as well. So Drosophila is my model organism. So Drosophila melanogaster is the broadly studied fly species. I do work on other species of flies in my lab too, on a variety of different evolutionary questions, but for the purposes of this talk, I'm focused on Drosophila melanogaster. What's important about it is its insulin signaling pathways are highly conserved. So across all metazoans, insulin signaling occurs, and it's highly conserved with many orthologous genes and enzymes in the pathways that parallel each other. So we can model it with mammals well enough, but we can also do a lot to model it with invertebrates, including C. elegans and Drosophila. So also of importance is that in the Drosophila genome, at least 77% of the known genes on the online Mendelian inheritance of man database have cognates in Drosophila. So we can really do a good job of modeling many aspects of genetic variation and genetic disease in Drosophila relative to humans. In terms of some of their physiology, humans and flies have a lot in common there. So just as one example, the fat body of the fly is a composition or a collection of the equivalent of adipose tissue, liver, and innate immune system in humans. So there's organ parallels as well. And flies also have hearts, which I'll get to in a moment. So how do you actually measure a genotype by environment interaction effect, if that's what you're trying to understand? So your challenge is that when you get to a wild population, you see lots of phenotypic variation. Here I'm representing that with different colors. So that variation could be environmental, it could be genetic, you just really can't tell looking at a wild population. But what you can do with something like flies and some other model organisms is you can bring them into the lab, different genotypes, different samples, and establish inbred genetic lines from which you can assess the genetic contribution to the phenotypic variation. So when I can compare across these inbred lines, I can make a robust estimate of the genetic variation in a given trait. But because of that environmental heterogeneity that may also be occurring in the wild, I can systematically test for that environmental component to the variation in a lab environment by systematically placing these different genotypes across these different environments. So I can test every possible interaction between genotype and environment, and thus figure out what the interaction effect really is in this system. So this is the basic approach I use in all of my major experiments. So this is one of the earlier studies that I did where established this research program where I was looking at 4 different diets in Drosophila. I had a wild population of, or a set of inbred lines from a wild population of Drosophila, about 150 of them. And I raised these flies across 4 different diets. So what I call a normal diet, which from the fly perspective is a balanced lab diet, a control diet, which had lower sugar than the standard lab diet, a high glucose diet, and a high fat diet. So supplementing the normal diet with either more glucose or more fat. And then I went on to measure a series of phenotypes associated with metabolic syndrome. So weight, blood sugar, lipid storage, and then characterize survival traits. And then out of that original 146 lines, I also characterized for 20 lines the metabolomic profiles by GCMS and whole genome expression profiling. So this is the early days of metabolomics. This is the last time I actually touched a GCMS myself when I ran these samples. I don't trust myself to do it anymore, really. But this is some of that early data that is driving the overall field. So in general, for any phenotype, in this case, I'm just modeling the phenotype of body weight, we can expect several possible types of outcomes in response to variation across diets. So each line here on this figure is representing a different genetic line, or line that's been isolated from the wild. And then the phenotype is represented at these points relative to the different dietary treatments. So we might have a genetic line like this black one that does not gain or lose weight relative to its diet. So it's unperturbed by its diet in any way. And I know a few people that seem to be like this. So there is that genetic potential out there. There also could be ones that say lose weight on a low calorie diet and gain weight on a high calorie diet, or still others that might react to a specific type of calorie excess. So in this case, the pink line is gaining weight specifically on the high fat diet. So these are all possible outcomes from this type of analysis. When I look across the actual data, in this case, summarizing across the environments, just looking at the genetic effect on body weight, across these 146 lines, there's huge variation in the amount of the genetic contribution to body weight in these flies. And the spread that we have here, if you scaled it to humans, is comparable to what we see in humans. So there's this huge variation in human populations in the genetic component of body weight as there is in flies. These little ones highlighted up here are to emphasize these are 2 standard lab strains that many labs focus on doing Drosophila work. And I highlight them here to emphasize that our lab flies, our traditional lab flies, are not necessarily particularly representative of what's actually happening in the wild for this species. So now when we look across the different diets, we see this spaghetti diagram mess. My postdoc advisor called it graphical vomit, which is a fair assessment. So basically this is showing that for all 146 different lines, basically every line had a different way of reacting to the environmental conditions. So I've color coded them by similarity in their their basic reactions. So for example, these red lines down here tend to maintain relatively low weights and be relatively unperturbed by the differences in diet. Whereas there's others that can be strongly influenced by the diet, like these green ones, where they happen to lose weight on a high fat diet. Again, these are the 2 canonical lab strains for comparison. So basically, now that I have this graphical vomit, I can statistically analyze it and tell you that 19% of the variation observed in this population for the phenotype of weight is genetic, strictly genetic. 2% is dietary. So I was really, really surprised by this number. I was expecting the dietary component to be comparable, if not larger, than the genetic proportion of the variation in the population. So this was a huge surprise. And then what is further surprising is that there's a very substantial genotype by diet interaction effect. So about 12% of the total variation is due to that interaction effect. So what that means is that it's not the diet alone that we should be analyzing when we're thinking about these traits, but more specifically the diet and how it interacts with the genotype is going to be driving what we observe in natural populations. One other small note is that there was significant decanalization on the high-fat diet. I haven't labored that idea, but the concept is that when a population moves into an environment that is not well adapted to or has not experienced previously, there can be a release of genetic variation. So cryptic genetic variation can be released. And we observe that again in this population, specifically on a high-fat diet. So now going on to where we analyze the metabolomics and the gene expression profiles for a subset of these lines, we replicated the basic patterns of the amount of variation due to genetic effects in the gray bars here. Dietary is a smaller portion, and then a larger proportion that is the interaction effect between genotype and diet. And then when we look at the expression profiles, we find that the expression profile in these flies has a very small dietary component as well, and a large genetic effect and relatively large genotype by diet interaction effect. In contrast, the metabolites have a much larger impact of diet, and perhaps that's not surprising. You might expect that the dietary contribution to the metabolomic profile would be a more direct reaction to the environment or the diet. What was depressing to me about this finding was that the correlation between the expression and the metabolomic profiles, principal components, were not correlated. So basically, I could not use anything about the expression profiles to predict anything about the metabolite profiles on an overall scale across the data set as a whole, and vice versa. So they seem to be decoupled at the level of the full data set. So this again runs sort of counter to my intuition going into it. I was expecting there to be some kind of sort of linear relationship where I could watch how things pass down the endophenotype progression, and that did not hold out when we got to actually looking at the relationship between these two endophenotypes. An additional level of depression or fascination, depending on your perspective, was that when we looked at the relationship between which genes and which metabolites correlated with the specific traits we were measuring. So here I'm just showing you body weight, but remember I measured a bunch of different phenotypes, that the specific genes and or metabolites that correlated these traits tended not to replicate across diets. So what that means is that if I measure, say, a gene that changes expression levels in correlation with my diet when I'm eating a high-fat diet, and that becomes an interesting gene to me, that's unlikely to be particularly useful in predicting what's going to happen in another dietary condition. So again, this highlights the need to really think about the environmental context of the population you're analyzing or the individual you're analyzing. You can't really generalize across different environmental conditions particularly well. It turns out, though, that there was a little bit of useful information buried in there in terms of predictability. And where that was in the metabolite, the second principal component of the metabolite profiles. And it turns out that that one was actually a fairly good predictor of METAS phenotypes. So the metabolome as a whole wasn't necessarily a particularly good predictor, but when you think about some of the major dimensions of variation, there is some predictability. So in this case, this is looking at body weight across the 4 diets and those metabolomic profiles. When we look at the loadings on that first or the second principal component, the metabolites that are especially prominent in that include branched chain amino acids, which is consistent with what we've seen in the literature in general for obesity and metabolic syndrome traits, that there's often a correlation with elevated levels of branched chain amino acids. And then this group, which was particularly interesting to me, so L-DOPA being the precursor to dopamine, and then this one, N-arachidonyl dopamine, which I had never heard of and actually did not realize was in there initially. So again, this was the early days of metabolomics. Our databases for comparison were somewhat limited. So when I did that initial characterization, it came back to me as THC, so the psychoactive compound in pot. And my initial reaction at the time was to freak out. I was terrified that somehow there had been some kind of systematic bias introduced into my years-long study by the partying habits of my undergraduates that were working with me at the time. Somehow THC had gotten into my experiment. Fortunately, somebody a little more rational suggested that perhaps it was something else, like an endocannabinoid. And when I actually dug in and ordered all those standards, I was able to confirm that it was this particular endocannabinoid. So endocannabinoids are important for regulating a lot of functions in our physiology, including appetite. So in the same way you can get munchies when you smoke pot, this particular endocannabinoid might very well be modulating the appetite levels of the flies and thus driving some of the variation we're observing in the phenotypes relative to those metabolite levels. The other really interesting thing that came out of this is that I mentioned that flies have hearts. Their hearts are homologous to human hearts. And we can actually measure aspects of heart health in the flies. So this is image down here on the right is what a normal, healthy young fly looks like with a consistent, regular heartbeat. As it ages, it develops arrhythmias. So, and the degree of arrhythmia can vary genetically and with lots of other characteristics, lots of other factors that affect the health of the fly. So what we did is we measured the arrhythmia of these genetic lines when across these different diets and asked whether those arrhythmic patterns were in any way correlated with any of the other things we measured. And what we found is that this arrhythmia index, the development of heart disease in these flies, was strongly correlated with this second principal component of the metabolic syndrome. So an additional thing to note here is that all the phenotypes I've talked about thus far have been measured in larvae, so early stage development, whereas this particular phenotype is measured in adults. And so what this means is that larval characteristic can be predictive of what the adult's disease condition is going to be when it comes to something like heart disease. So this is actually really encouraging, suggesting that there can be useful data pulled from early stage metabolomic analyses that can be helpful in guiding and predicting treatment going forward. So I mentioned that when I looked at the metabolome as a whole, there was relatively little correlation between the metabolites and the genes, and it was sort of frustrating that I didn't see any logical connections there. And so we adopted an approach where we looked at whether there was structure within the metabolome that could help us identify relationships to functionally active genes. So basically what we took our metabolomic features, clustered them based on correlations, so doing this hierarchical clustering. Then we took a cluster and ran principal component analysis on it, took that first principal component, which we can call the eigenmetabolite, and look for correlation between that principal component and the various genes that we had also profiled in these same samples. And once we identified any genes that were correlated with that, we can then analyze them for potential functional enrichment and look at correlations with the actual phenotypes like the body weight that we had measured. So this is what that clustering ended up looking like, very simplified. So for example, there's this cluster up here that had a bunch of fatty acids in it. This cluster down here had a bunch of amino acids in it. And when I looked at the correlated genes, some of these clusters had many correlated genes for expression profiles. Others had virtually none. So this partly indicates why there is that breakdown of relationship between the expression profiles and the metabolomic profiles. Some aspects of the metabolome were well correlated with gene expression and others were not. So there's subtlety within the structure of the metabolome. In terms of looking at enriched terms, things like the mitochondria, so generation precursor metabolites in the TCA cycle were associated with this particular cluster. That was an enrichment for those terms among those genes. And then we can say which of the overt phenotypes, like body weight, were correlated again with these various metabolomic clusters. And so we found that some of these clusters, like this one, correlated with several phenotypes. So in this case, blood sugar, lipid storage, and body weight. Others only correlated with a single phenotype, such as body weight. Or in this one, this particular amino acid group, in addition to not correlating with any genes, also didn't correlate with any of the overt phenotypes. So again, we can partition out these large-scale systems biology data sets to understand the bits and pieces that are most strongly associated with particular traits. The other thing at the time that I was very excited by was the fact that there was this clustering, in this initial clustering, of chemically similar metabolites based just on correlations. So that was encouraging to me that the correlational structure had some grounding in reality. So in another study where this particular data set came from Metabolon, and we were looking at whether we could differentiate between the two different dietary treatments in the metabolite profiles of the flies. So a similar design to what I showed you earlier, except that we just had two diets in this case, a normal diet and a high-fat diet. And partly because Metabolon has a different library and different capabilities than what I did in my initial analyses, we were finding different metabolites as being important or involved in this differentiation among diets. So among the things that showed up there were things like short chain fatty acids were important in differentiating between the diets. And this group of metabolites here were especially interesting because they brought up the possibility that there might be some omega fatty acid oxidation involved in response to a high-fat diet. So when we look a little more carefully at the fatty acid oxidation potential pathways and how those were reflected in our metabolomic data, we saw this excess of medium chain fatty acids and an excess of products associated with omega fatty acid oxidation. And then we had an elevated level of TCA cycle metabolites in the context of a high-fat diet. So all that was going on while the carnitine shuttle was not showing any change. And so what we suspect, though we have not functionally confirmed, is that on a high-fat diet, there's a shunting of fatty acids away from traditional beta fatty acid oxidation towards omega fatty acid oxidation before pushing it back out to the TCA cycle. So the shift to this generally less preferred omega fatty acid oxidation is invoked in the context of a high-fat diet. Okay, so I think I've at least partially convinced you that there's genetic factors and there's environmental factors and they interact. One of the next obvious questions is, what are the genes that can drive that genotype by environment interaction effect? What's actually underlying those traits? So there are a number of resources available in Drosophila for genetic mapping. The data I'm going to show you now is based on this particular resource, which is the Drosophila synthetic population resource. So if you're familiar with the collaborative cross in mice, this is a conceptually similar design where there are several founder strains to the population that get crossed to each other and then subsequently inbred again, such that each inbred line is a random combination of the founder's genotypes. And it turns out that this is a very powerful genetic mapping tool. Makes it, it's just an extremely efficient way to get at gene level analyses in profiling. So what I did is I took these recombinant inbred lines, I raised them on the normal diet and the high-fat diet, and then measured their phenotypes and asked whether there was an association, genetic association, between the phenotype and the genotype for those given lines. So what we found is that, yes, there is. So this particular graphic is looking at the body weight of male pupae, and each little red dot is the maximum point of statistical association with that particular, with that trait in that position in the genome. So we found a number of loci that were associated with each of the metabolic traits we're looking at. And we were also able to map the ones that interacted with the variation in diet. So the loci, we could independently map the loci that were responsible for the interaction effect from those that were responsible for the main effect or the primary genetic effect of the trait. And what was interesting about this is that they don't overlap. So the loci that are responsible for the main genetic effects are different from the loci that are responsible for the genotype by environment interaction effects. So again, this was not intuitive. In quantitative genetics in general, the assumption is going into it that first you have to have a main effect before you bother even looking for an interacting effect. But we had sufficient power in the study to look at whether interactions could be playing a role independently of the main effects, and indeed they are. And in fact, most of the interaction effects are independent entirely of the genetic effects. So the graphic down here is just graphing out physically where those significant QTL were for the genetic effects in blue and the genotype by environment interaction effects in red across the various phenotypes that we measured. So basically you see that there's little overlap between the significant QTLs for the genotype and environment interaction effects. We also had the power to estimate epistasis. So epistasis is when two or more genetic loci interact with each other in a non-additive way to produce a trait. So again, we had some pretty high power in the study, and we were able to estimate the interaction effects throughout the whole genome, and able to associate those interaction effects with particular loci associated with particular traits. So basically, we found a whole lot of epistasis. So the take-home message from this little bit is that that large amount of epistasis is important for driving the genetic variation we observe, but it's extremely difficult to actually measure, especially in a population like humans where you can't do the controlled process. So it's important to remember that the estimation of genetic factors that you can do in say, a human population are limited to those sort of main effects, things that are not due to massive interactions. But there's a whole lot of additional complexity to the architecture of the trait going on under the surface due to this interaction among loci. So if you're having trouble wrapping your head around what these different types of QTL might look like, this is a little graphic I used to try to explain it. So you can imagine that there's a genetic effect. We're looking at two genetic loci, the A locus and the B locus. And here I'm representing the phenotypes as the white, gray, and black as just different phenotypes that this organism might have based on its genotype. So in this first example here for the genetic locus A, as you add the big A alleles, you change from white to black. But the B locus does not influence this particular trait. If you had a main effect genetic locus, you'd expect to see the same pattern in both dietary treatments. That is, the change in phenotype would be consistent between the two dietary treatments. But if you have an interacting effect, you might expect the phenotypes to differ relative to the A locus genotypes in the different diets. So what we were finding is that the loci that did this kind of thing were different from the loci that did this kind of thing. And then if you want to wrap your head even further around it and think about epistasis, when there's epistasis, it means that there's two or more loci interacting. So for example, it could be that the black, gray, and white variation can only be observed across the A genotypes in the context of the little b genotype, little b. So that would be an example of an epistatic trait. Whereas if you don't have the little b genotype, everybody looks like that big A phenotype. Or again, if you bring in a dietary interacting effect, it's possible that the nature of that epistatic interaction changes based on the diet. So basically we see all of these kinds of loci mapped in our study, and just makes things difficult to interpret in terms of figuring out specifically which loci are the most important ones. We have done gene ontology enrichment on these groups of genes, and we do find a number of genes that are logical ones to be associated with metabolic functions. One that was particularly interesting to me was this cluster of microRNAs that are involved in regulating the hedgehog pathway. So this is an area for subsequent functional follow-up. I'm really interested in trying to understand how this group of microRNAs may be playing a role in these traits. And many of the genes are associated with pathways that regulate growth and development, especially neurodevelopment. So that played a big role in the groups of genes that showed up in these studies. So we've talked about the main phenotypes, things like weight and triglyceride storage. Now, what about those metabolomic phenotypes? So in the same study, we also collected a number of samples that we then sent off to Oliver for profiling. We were able at that time to profile about 421 metabolites, 170 or so with confirmed chemical IDs. And just to think about what we're going to see in these subsequent images, there are several different types of things that could come out of this type of analysis. One possibility is there might be a simple genetic architecture for the relationship between a metabolite and a gene. That is, one gene modifies the levels of one metabolite. We might find situations where there's multiple genes, so that influence one metabolite. So I've been calling these hub metabolites. And there might be situations where there's one gene influencing multiple metabolites. So that could be a hub gene. So when we actually look at the analyses, and I'll walk you through this, it's a little bit daunting. We basically see all of those types of scenarios playing out. So the color coding here is that red are main genetic effects, blue are genotype by environment interacting effects. So again, we have QTLs that differentiate between those main genetic effects and the interaction effects. And if you squint at this, you'll see that they're largely non-overlapping. So again, similar to what we found with the main phenotypes that we're measuring, the metabolomic phenotypes generally don't have the same QTL underlying them when you're looking at main effects versus interacting effects. What's being graphed here across the x-axis is the physical position in the genome for across the different chromosomes in the Drosophila. The y-axis is the metabolite index, so just the which particular metabolite we are measuring. And then if there's a dot, there's a significant association between the genotype at that position and that particular metabolite. So we had some situations where there's what I might call a simple QTL, where there's a direct relationship between one gene and one metabolite, and they seem to be operating independently of any other genes or metabolites in the system. There's other situations where one particular metabolite, in this case, is associated with many positions in the genome. So it's a hub metabolite. And situations where there's hub genes, where there's one gene that's impacting a number of metabolites. If we drill down into one of many examples of these situations where we have the hub genes, so one gene affecting multiple metabolites, this is just one example of when we drill down and look at what's actually under that peak in the genome. That under that particular peak in the genome there happens to be something that overlaps with pyruvate kinase. So this particular metabolite is one that's shared between glycolic acid and citrulline. And it turns out that pyruvate kinase, being a rate limiting step in glycolysis, is a logical point at which you might see a genetic association between both of these metabolites and a position in the genome. So if that gene is coding for pyruvate kinase, and that particular, the genetic variation in that pyruvate kinase that influences the efficiency of this particular pathway, that would explain the correlation in the genetic association between the glycolate and the citrulline. Okay, so I've been thinking a lot about how do we actually break down all this complexity into something that's manageable, something we can actually wrap our heads around and potentially develop testable hypotheses about. And so one realm where I've been exploring this is in graphical analysis of relationships among metabolites or genes, a variety of different options. So in Gaussian graphical modeling, which is one of these strategies, the idea is that you might have this group of 5 metabolites, perhaps 2 of which we don't know the chemical ID for, but we know something about how they're correlated to other metabolites. In this case, known metabolites 2, 5, and 4. That correlation structure is not particularly helpful because it ends up being everything is correlated with everything else, generally speaking, in these types of data sets. It's really difficult to pull out the most salient pieces. But you can, in Gaussian graphical modeling, apply a conditional dependence. So basically, given the measurement at one metabolite, is there still a dependency between two other metabolites? And when we do that, we can actually look at what are those important, more likely to be functional relationships between metabolites. So in this hypothetical example, the correlation that metabolite 2 has with metabolite 3 and metabolite 5 is is conditionally dependent on metabolite 1, such that if you can account for the relationship between metabolite 2 and metabolite 1, you have explained the relationship between metabolite 2 and 5 and 3. And it turns out that these tend to replicate biochemical pathways. So in this study, we were looking at, again, this is going back to that metabolon data set. And we were looking at the correlations, the conditional dependence among metabolites, both on the normal diet and the high fat diet using Gaussian graphical models. And if you see a little blue dash here or red dash here, that's indicating that there was a significant conditional dependence between those two metabolites. Then we were able to overlay these two networks, and from that extract out groups of metabolites that had a relationship that's logical, consistent, given their chemical compound and the relationship to each other. So there's this particular data set had a bunch of dipeptides in it, and we had these clusters of dipeptides that came out as being important in driving the variation in these under these two dietary conditions. We have some phospholipid subnetworks, some fatty acid subnetworks. When this is just an example of how we can use that Gaussian graphical modeling to actually extract useful information. So again, in this case, we're looking at the data set as a whole. And if we do the correlation level with a fairly stringent correlation cutoff, we basically see just this hairball, this fairly useless set of data. But when we're able to apply the Gaussian graphical models, we see a much simpler structure. And coming out of that structure, we see logical relationships. So in this case, this is fatty acid synthesis. I consider this now to be my positive control for whether my statistical approaches are extracting things that are useful, because I often end up seeing this, and if I don't see this, I'm worried. So basically, that transition from C8 to 10 to 12 is replicated in the conditional dependence in this Gaussian graphical model. And so going back to that dipeptide clustering, so again, if we did the correlation alone, we just end up with this hairball. It's, to me, uninterpretable. But we can start seeing logical relationships among these dipeptides when we actually do the Gaussian graphical model. And what that does is it gives us testable hypotheses. We now can suggest that there should be some link, say, between these two dipeptides from a mechanistic perspective, and we can start drilling in to figure out what that link might be. And that's going to be especially helpful when we have situations like this, where we have unknown metabolites or unknown metabolite associated with known metabolites, where we can start using their association with known metabolites to start diagnosing what they might be and how they might be connected mechanistically to the larger group. Okay, so I want to briefly talk to you a little bit about some of the other environmental conditions that my lab is thinking about. So my ultimate goal in my lab group is to be able to model all of the major environmental influences on obesity and diabetes, but using our fruit flies. So one of those, in addition to diet, is exercise. Exercise is clearly plays a major role in many of these phenotypes. And so what we've done is we've invented a way to exercise the flies. So fruit flies live in these little vials, at least in the lab environment. And they exhibit a trait called negative geotaxis, which is basically if they're at the bottom of something, they want to go up. So what we developed is what we call a tread wheel, which is this machine that slowly rotates vials And as those vials rotate, the flies crawl up, and then eventually the vial gets flipped all the way over, and they are crawling up again because it keeps flipping. So basically, they just keep walking, and we can induce them to walk for long periods of time using this approach. As our control, we confine the area over which they can move during that rotating portion of the experiment. So basically, exercise works. You can exercise flies. So this is some of those phenotypes that we commonly measure: triglyceride, weight. We also measured protein and glycogen storage in this particular study. And so, for example, triglyceride levels are reduced significantly in the context when flies are exercised. We also see an improvement in performance. So in this case, climbing performance, which is basically how quickly can a fly climb from the bottom to the top of its vial. And we see a significant improvement in the performance of the flies under these exercise conditions. We've then gone on to look at whether there's dietary interaction effects that work with that exercise. And we found that exercise interacts both with genetic lines, so there's genetic variation in how effective exercise is, and with sex. So the different sexes of the flies, males and females, respond differently to the different treatments of exercise. We find that genes associated with mitochondrial function, so fission fusion associated genes, along with a few others that in that general group, are showing significant genotype by exercise interactions. So we can functionally identify some of those loci or some of those genetic factors that could be playing a role in this. And we also have found that if we feed the flies as larvae a particular diet and then exercise them as an adult, so in this case we are looking at whether we could potentially model juvenile diabetes or juvenile obesity, and whether interventions in adults could be effective. We find that, again, there's genetic variation for how effective adult exercise is in mitigating larval diet. So the effect of exercise varies. So for example, this red line here is one genotype where it happened to decline in performance on a high-fat diet, but when it had a high-fat diet and exercise, its performance improved. So it's an example of an interaction effect between the larval diet and the adult exercise regime. So another area where my lab is working in is looking at larval bacterial communities. And we are especially interested, a lot of work has been done in gut microbiomes and thinking about how those influence obesity traits, among other things. And one of the challenges with fruit flies is that most of these studies have been conducted in a controlled lab environment. And wild fruit flies live out in rotting fruit. They are completely surrounded by and consuming all sorts of little critters, microbes of various sorts. And so we suspected that the microbiome, the role of the microbiome was going to be very different in a natural system than in a lab system. So in this study, the basic design was we put out, sorry, where's that? Oh, there it is, all right. We put out a bunch of peaches in my backyard. This is my daughter. Put out a bunch of peaches in my backyard and allowed them to rot in the middle of the hot, humid summer here in Alabama, and then collected them 6 days later and froze them. And then we've been using those peaches as the basis for our wild rotten diet ever since. So we had the basic dietary treatments then are rotten peach, autoclaved rotten peach, where we've killed off the live microbes, and then comparing that to a regular lab diet. And then within each of those dietary treatments, we've sterilized the larvae. So it turns out that it's surface sterilized, remove the microbes that the larvae might otherwise bring with them from their mothers, versus ones that are non-sterilized. So it turns out there's a lot of vertical transmission of microbes from mothers to larvae in the process of the egg laying and the larval hatching, where they eat the egg shell around themselves. So we wanted to control for that factor. One of the important things we found is that the presence of maternal microbes, that is those vertically inherited microbes, played a big role in shaping the microbial community or bacterial community. So for example, here on the peach diet, which is what's over here on the left, the sterilized, that is the ones that did not inherit maternal microbes, and the ones that did inherit maternal microbes are substantially different from each other. Further, these peach diets, natural peach diets, are significantly different from the diets that were either the lab diet with this regular, that's what the R is standing for there, or the autoclave diet on the peaches. So basically the natural conditions that a fly experiences in the wild in terms of the microbial environment it's exposed to is very different from what it's going to experience in the lab. And thus we need to be cautious about interpreting lab-based studies in a generalized way, given that there can be these dramatic differences. There were also genotype by diet interaction effects in this study. And we found a number of correlations between dominant taxa in these microbial communities and metabolic traits, and many of those were consistent with what's been seen in the literature in mammalian systems. We also looked at whether supplementing the diet with fat or sugar influenced or interacted with the presence of those microbes. And just I'm going to focus on the left here where we supplemented the diet with fat. And then if we look at the separation of the communities on the different diets, so this group here is the natural peach diet and the lab diet versus the autoclave peach diet, it turns out that if you kill off all those microbes in the food in the autoclave peach diet, you have a very different community resulting in high fat conditions. But in contrast, when you're on high sugar conditions, there's not that big differentiation. So the role that the gut microbiome is playing in developing the communities in these different, under these different dietary conditions is dramatically different. And we've also found then that the bacterial species that correlated with particular metabolic phenotypes vary with the diet and vary with those maternal microbes. So whether a particular bacterial species can be consistently associated with a given phenotype is highly conditional on the environmental condition under which that microbe is and diet that that host is experiencing. So the take-home messages here is that flies are great models. Genotype by diet interactions are a substantial contributor to META-S variation. Metabolic profiles have predictive power in META-S phenotypes. Quantitative data can be used to categorize unknown compounds and hypothesize new pathways. Loci for genetic and genotype by diet interactions are different. And there are many additional environmental factors that we can successfully model with Drosophila. So the just last little thing I wanted to bring up before closing for questions is another big project that I'm involved in, which is the Genomics Education Partnership. So I've taken over as director of this program as of about 3 years ago. And it's a really exciting program that provides course-based undergraduate research experiences. So there's extensive data in the educational literature that students doing actual research is one of the very best ways to both teach them science and retain them in the scientific field. And so this particular project reaches over 2,000 students a year. That number is growing dramatically this year under COVID conditions, with currently about 160 institutions that are involved. The GEP team provides the resources for conducting this research. Everything is GUI-based, web-based analysis, genomic analyses. So there's no barrier, like scary command line barriers to undergraduates who might otherwise be interested in the concepts and ideas that they might be learning about in a comparative genomic type analysis. So we use these comparative genomics to develop high quality gene models in species that have been under-annotated. So that's the basic project that the students are doing. And then as a group, we take all of those high quality gene models and analyze them in a meta-analysis to understand something about gene and genome evolution. And all of our assessment data shows consistently that the students really do gain confidence in their ability to do science going through this process. So what I wanted to highlight here is the scientific project I'm leading within the Genomics Education Partnership, which we call the Pathways Project. So there are multiple scientific projects occurring using this basic formula, but in my case, we're currently focusing on insulin signaling. So basically, we have those draft genomes. We develop genome browsers and corresponding tools to allow the students to work with those genes. In my case, I've identified a pathway of interest, starting out with the insulin signaling pathway. We look at those orthologs, putative orthologs, between the reference species, which in this case is Drosophila melanogaster, and other species. Students, faculty claim projects, and their students produce these models. We have at least 2 independent models for each gene. We then reconcile those models. And once we have those reconciled models, those get published both through something called micropublications and also deposited into public databases, becoming available to the scientific community more broadly. And as I mentioned earlier, ultimately analyzed in a meta-analysis to understand something about gene and genome evolution. So the reason I bring this up here is that for 2 reasons. One is we're looking for new educational partners. So if there are faculty or future faculty out there who are interested in incorporating some hands-on real research into a genetics or genomics class, this might be an excellent way to do it. We provide all the training and the resources for it. We're also looking for scientific partners. So if you have a group of genes in an genomically under-analyzed species group, that's something that we could really help with. If we have a clear set of genes and we can construct the tools to help with annotating those genes if you're interested in something about the evolution of particular, say, biochemical pathways. So with that, there are lots and lots and lots of people who have contributed to this research and a number of funding sources. And I think I'm ready to take questions if there are any.
Thank you very much. Arpan, do you want to take over?
Yes, I'd love to. Thank you so much, Dr. Reed, for that wonderful talk. I have a couple different topics and questions that have been in the chat room. And if you as audience have more questions, please submit them in the chat box. There are a couple questions on genetic variation. You spoke about how genetic variation is only accounted for the Drosophila genes. How do you account for microbiome diversity? And is it possible that they, trump those genetic differences?
So yes, so there's a couple different things going on. One is that variation in the microbiome can be driven by genetic factors of the host. So that's one of the things we've analyzed and find evidence for, that the microbiome varies in reaction to, across different host genotypes. But also certainly the availability of particular microbes or the composition of a particular microbial community may indeed override the genetic propensity of an organism. And so part of what we really want to understand in the work that we're doing is figuring out how do those different pieces interact and under what conditions does one or the other predominate. So yeah, I'd say, does that help?
Yeah. And there was also another question. You spoke about there being 30% of the total variance was explained by either the diet or the genes, or maybe like the interaction with the environment. So wouldn't that mean that the rest, or 70%, is due to other causes in those variants? Have you ever considered maybe the dark matter of variance, which is something that Oliver coined here in the chat box, and maybe could be bacteria?
So the answer, there's several layers to that answer as well. So yes, there are other factors in that 70% unexplained. So some of it's just going to be completely uncontrollable, unmeasurable environmental variation or random variation. But yes, there might be microbial contributions, but the other major thing that's likely contributing there that we cannot capture with those particular types of statistical analyses is all that epistatic variation. So I showed you those big hairballs of epistatic interactions. Traditional linear genetic analyses can't capture variation that's due to those epistatic interactions. So the genetic component that I can characterize is the heritable component, the part that is due to additive effects, essentially. But I cannot, with that particular approach, characterize the proportion of that variation that's due to epistatic effects. But certainly there are other things that are contributing.
Yeah, I mean, there's so many factors, and it's, you know, it just like how you were showing that hairball diagram, just taking it apart definitely takes time and expertise. There were many questions about correlation. It- More specifically, one of our grad students has been working with mouse plasma across many platforms and found that there was very little impact of those metabolites on body weight. She wanted to know if you found a large correlation of the metabolites with body weight, because that's something that she didn't find.
So yeah, for some of the metabolites, yes, there was a strong correlation. So especially those that, going back to those, that principal component 2, the ones that were in that group, so the branched chain amino acids and some of the neurotransmitters were strong correlates with body weight.
Excellent. I hope that helps Jenny with her own investigations.
Yeah, and I'm happy to talk more offline about that too.
Oh yeah, that's wonderful. Yeah, that's the great part of Zoom is we can continue to chat.
Right.
There was definitely an interest in how you performed your correlations. Did you know, like, what specific analysis that you did? Maybe did you do a lasso regression? Is it possible that you did multiple testing in terms of corrections? And what was the typical strength you used for correlation? Was it something that was normal or stronger in terms of the strength of your correlation?
So, yeah, so in terms of multiple testing, yes, we provide, use both. We've looked both at FDR and at Bonferroni corrections for multiple testing on all of those analyses. And of course, when you provide a more stringent threshold, there are fewer significant correlations, but the patterns don't change. I mean, so the overall structure of the data doesn't change in terms of what seems to be important or what the patterns are. Most of the basic correlation analysis are just standard linear regression. And when we got into the Gaussian graphical models, that was a graphical lasso. So.
Yeah, those networks were really nice. I liked how visually it was nice to see and to understand as well. There was some questions focused around like the possibility of multiple genes, specifically at the F75 level, were your loci pointing at a single gene or multiple genes?
So the locus was, any given locus was generally overlapped with only a single gene. Occasionally they're a little wider and included more than one gene within that locus. For our high throughput analyses, we went with the gene that was closest to the peak, just because it just became a mess with so many significant associations. But there are certainly possibilities that when you look under a given peak, that there are more than one potential candidate. And that would take drilling in on a case-by-case basis to identify whether the peak, the most significant association position, the gene under that is the best explanation for that particular genetic association.
Great. There was also a question on gene interactions and if it could be possibly caused by gene folding or transcription factors.
Mm-hmm.
Could you speak a little bit more about that?
Yeah, so epistatic interactions are driven by a lot of potential things. One of the most intuitive ones is transcription factors or regulation, gene regulation. So if you have one transcription factor protein that comes in and regulates another protein, if there's genetic variation either in the transcription factor or in the binding site for that the protein that it regulates or the gene that it regulates, that can lead to an epistatic interaction. So a lot of those interactions probably are in some way regulational like that. Certainly any other time that you have multiple components that have to work together, protein complexes or cooperative folding or anything like that that has to happen with multiple players coming from different places in the genome, then you have the potential for those epistatic interactions.
Oh, That's very interesting. There's some other different types of questions that kind of don't have a general theme, but very interesting. One was proposed by Dr. Barnes here about autoclaving the peaches, and if that would change the diet, thereby controlling the microbes that can survive in the fly. Essentially, how would that impact the food that they're eating and the microbes that live on that?
Yeah, so we did a lot of thinking about the right way to try to control for the microbes in those peach diets. And I don't think that any way was perfect. So with the autoclaving, we do modify the nutritional composition, sort of caramelizing some of the sugars and things like that, that do make some changes to the nutritional composition. On the other basic strategy would be to load it up with antibiotics, but then there's a whole lot of other problems that can propagate from using antibiotics in that kind of study. So we opted for the autoclaving partly because we were doing this high throughput approach as a first pass. What so there are probably differences between the autoclave diet and the peach diet that are not related to the microbes, but due to the fact that it was autoclaved and some nutritional change. But when we add in the variation of whether or not the larvae were cleared of their parental microbes, we see a correspondingly similar effect in the autoclave diet and the non-autoclave diet. So the addition of those other microbes on the autoclave diet still modify the way the flies respond to the diet in a way that's consistent with what we see on the non-autoclave diet. So there's it's complex, but there's still evidence that the microbes themselves are playing a role on the in the autoclaved versus non-autoclaved diets.
Was there a specific reason why peaches? Is there, a preference for the flies or?
Right. So since we were going to be, well, I do backyard field biology. so we all the Drosophila in the Southeast love peaches. They're certainly not their native host. Drosophila melanogaster from Africa, and it eats other things there. But it's quite experienced with eating peaches around here. And if anytime we go out collecting wildflies, a peach orchard is about the best place you can go. So we were able to get lots of organic peaches here in Alabama that were readily available. We knew that the flies would like them, and we knew that lots of other things that would also be contributing to the rot would like them. So that was the logic. We could find that there'd be other substrates would produce different results, but we opted to go with peaches just because they were abundant and the flies generally would like them in the wild. So.
Yeah, that totally makes sense. There's a question about the use of male Drosophila and why maybe you used only male, or maybe did you use female Drosophila? Did you account for sex differences and gene-sex interactions? And maybe what did you find with that?
Right. So there are some situations where we only use male Drosophila, and that's a purely logistical thing because it's to sex males and females of the pupae. I especially early on when I was working on this, I was very confident in my ability to know a pupae was a male. I was less confident that I had successfully identified a female pupae, that it may have been a clandestine male pupae. So initially I focused on male pupae because I was confident they were indeed males, and I knew there are huge sex differences in the species. As we got further along and I became more confident, my trainees became more confident in differentiating between males and females, we started including the females as well in our analyses. And anytime you're looking at males versus females, it's almost like you're looking at two completely different genetic lines. The males and the females have some correlation with one another, but not much. And you really have to think of them as separate sexes. Anytime we are not able to separate the sexes from each other, we did it with large pooled samples. So if we had males and females together, say in the case of some of our larval analyses, we did it with a large number of larvae, thus minimizing the probability that there'd be a strong bias in one direction or the other due to sex in that particular sample.
Oh, great. Yeah, it's definitely something that, we've come across in terms of like our studies, the one that I mentioned previously. And just one last question out of curiosity. I know we're coming up at 10 minutes past the hour, but the GE program that you mentioned, that kind of, interested me. I was wondering if maybe you're extending it to metabolomics. If not, if you're finding partners for that, I'd definitely love to talk to you about that.
Yeah, so right now there's not a specific plan to expand to metabolomics, but I could imagine a number of ways that we could. The critical thing is being able to develop a packageable project that a student can do that then scales well.
Mm-hmm.
And so if there are some systematic rules that we can teach to students and then have them apply it to a particular scientific question, and then that can scale, that's the sort of thing that GEP does very well. And so just off the top of my head, I could imagine that perhaps interpreting spectral profiles or something like that, where you need to apply a series of potential rules, and that perhaps the brain power of an undergraduate who's been trained in those potential rules could help process those things faster than an automated system might. Perhaps I'd have to think a little harder about it, but yes, I would definitely be interested in talking about potential things along those lines.
Yeah, we're expanding and doing more, courses. So definitely would love to talk to you about that. And with that, I'll pass it back to Oliver.
Thank you so much, Aparna. It was a great overview of the questions. And thank you again, Laura, for spending the time preparing for the seminar and being with us today.
Yeah.
I wish we would be able to meet, and we will someday soon.
Someday soon, yes.